Thank you very much. That was a nice intro. So back to reality, normal biology. I feel a bit sort of humbled actually by constructive bio. How does this work? Okay, so we're doing lots of different things, but I want to talk about one specific thing, one of my favorite things, and I'm going to give you like the farmer motivation for why we're doing what we're doing. So these are 80 oncogenes, so mutations in these things definitely cause cancer. Inhibiting these things would definitely be good as a therapeutic strategy, at least temporarily. These are the ones which actually have licensed drugs against them. This is what those proteins are. So we are really good at inhibiting certain types of protein, and we are not so good collectively at inhibiting other types of protein. So this is the motivation for a lot of what we're trying to do, is to work out, provide information for how to inhibit or activate other types of protein. So most drugs work by they're allosteric. They go into the active site of enzymes and block them, which is great. The problem with that is a lot of proteins do not have nice active sites, which are pockets. Protein interactions, protein DNA interaction, protein RNA interaction, for example. So then another strategy would be to find what's called an allosteric site in a protein, so a regulatory region where if you bind in a small molecule, you could affect the activity, modulate the activity. Okay, and the problem is for most proteins, we don't know where the allosteric sites are, and we don't know if there are any and whether they overlap nice structural pockets. So allosteric, depending on who you believe, Jacques Monod did or did not call it the second secret of life when he came up with the idea, the concept. But it's sort of basically central to all of biological regulation because it allows the activity of a protein to be regulated by something happening at a distance on another site within the protein. This could be a ligand binding, it could be a post-translational modification, could be a non-natural amino acid, and for us it's always mutations because we can make mutations at scale. And allosteric drugs are really nice because they can be much more specific. So all kinases have the same active site. It's really hard to inhibit one kinase without inhibiting 100 of them. And it allows you to drug potentially proteins which do not have active sites, are not enzymes. So protein, lots of oncogenes just work by molecular recruitment. They have horrible active sites. Okay, and the problem is we don't know where the allosteric sites are, and so we sort of have the challenge of making methods to just take a protein and find out where are all the allosteric sites on the protein, which in a way is just to say, is to measure at scale energetic coupling between different sites on proteins. So where can I touch a protein, do something, and it has an effect at a distance, and where can you not do that? And to make this as big training data sets so that we can try and understand this thing, mechanistically and also learn how to predict it so we don't have to do any more experiments. So yeah, we've always used these ducks to illustrate allostery, and I was on holiday in Osaka with my son, this is Alex, trying not to think about allostery, etc. And we walked past this thing and this happens and ruined my holiday for an hour. So how do we do this? So what we want to do is we want to measure the activity of proteins, which could be an enzymatic activity, could be an interaction. And then we want to perturb the protein all over and see what affects it. And so we use mutations as the perturbation. You can put in, using natural amino acids, 19 different 20 different changes at each place, non-natural, of course, more. And then you have to have a functional assay. So I'll show you the example using protein-protein interaction, because every protein you're interested in binds something as part of its function. So it's a very generic assay. But we have other assays where this is for enzymatic activity, DNA binding, etc. Okay, so if you think about protein-protein interaction, there are two obvious ways that a mutation could cause a protein to stick less to another protein, to form less of a complex. One is what we're interested in, is you change the binding affinity, the binding energy. But most of the time what happens is you make a mutation in a protein and the protein is just less stable or it aggregates, and so you have less of the protein, and therefore, of course, you have less binding to the target. So when we make mutations in proteins, we need to distinguish between these two things. We need to deconvolve the effects of mutations on the binding energies, the stickiness, from the just the folding energies, the stability of the proteins. Okay, so this is a very general thing we deal with all the time in the lab, is that when you change sequence and you measure something in the cell, there's normally an infinite number of possible mechanisms that could cause that change. So this is a very simple system where we are just measuring the interaction between two proteins. What's the concentration of this complex? There is normally an infinite number of possible binding energy and folding energy changes that could underlie that. And by making one measurement, you can't work that out. You have to do a titration experiment, would be the classic way. Or the way that we do it is we combine together mutations and we make lots of different measurements to decouple these things. So in our experiments, we make experiments where we mutate proteins and we use different assays to measure, do they bind to something we're interested in? And then we have a set of assays which measure what's the concentration of this protein, how much of the folded protein is there around on its own. Then we fit models to it, which looks very complicated, but it's actually quite simple. And this allows us to do massive scale biophysical measurements. So in these little proteins where we make every mutation in every position, we actually measure or we infer the changes in folding energy and binding energy for every sequence change in the protein. So it's like doing biophysics, but using genomics, which is much more scalable, much faster. So all of these experiments work the same way. So we synthesize normally or mutate big libraries of variants in our proteins. Then we put them through selection assays. So here you might have 150,000 different sequences. We sequence them to count how many there are at the start of the experiment. We select and then we sequence them at the end of the experiment, and that change in frequency tells you in that assay how well does each of them perform. And these are wonderful assays. Once you start doing this, you don't want to go back to doing arrayed stuff or parallel stuff because everything is in exactly the same batch. So everything is together. You know, you're making 250,000 measurements with no batch effect. Okay, and so for the stuff I'm going to show you, this is doing experiments in yeast cells, so human proteins in yeast cells. And we use these two protein fragment complementation assays, which we really like because we benchmark them a lot now against in vitro measurements. So one of them, which we call abundance, PCA, this is your protein you're interested in. This reads out the concentration of the folded protein in yeast. Okay, so the protein is tagged with a part of an enzyme. The rest of the enzyme is there at high concentration. The concentration of this enzyme now controls the growth rate of yeast. So if your protein is stable, it grows fast. If it's unstable, it grows slowly. And the binding one is similar. So you split the enzyme into two fragments. If the proteins come together, you have the enzyme. If the proteins are not together, you don't have the enzyme. So there's loads of these assays, and our approach is we just test them and we put in big data sets of reference mutations, so mutations in proteins which have been measured in vitro, so we can actually benchmark exactly how well these different assays work for different types of proteins. Okay, and then the model fitting bit. So in these experiments, there's two computational challenges. So first of all, you have hundreds of thousands of sequences which differ from each other by 1 or 2 nucleotides, which is a slightly non-trivial analysis problem. And then we have to go from these mutation frequencies. So this is our package for doing that, boom sum. And then we have to go to these, taking the double mutants and the different measurements and inferring out the biophysical things. And this is this package Mochi, which uses neural networks, but that's just to make it fast. But we're fitting to the data very, very simple biophysical models. So these are the stupidest neural networks in the world. So they're the little toy neural networks where the edges mean something biophysically. And we just use the PyTorch, etc., to help the fitting. So here's 2 examples from this paper. So here's a little SH3 domain in gray binding to a peptide. And then the residues which are shown as highlighted, these are the positions in that protein where when you make lots of different mutations, it changes the binding energy. Okay, so it is the binding interface, which we knew from the structure, but that's good. And then there's 2 glycine residues just behind the interface where you can make mutations there and it changes the binding energy. So these are allosteric, but they're very close. Okay, so changing the, putting in side chains there or changing the dynamics does something. Here's the PDZ domain. So this binds this peptide in black. Again, the positions where lots of mutations affect the binding energy is the interface in red. But now it's more interesting. Now you have residues going to the back of the protein where mutations on the back of the protein by some mechanism unknown, change the affinity on the other side. Okay, so this is now quantifying the allostery. Does not tell you the mechanism, it tells you that it's happening. So I'll show you a more interesting example. So that's doing on small proteins where we had lots of reference measurements to benchmark things. This is now making an allosteric map of an important protein. So this is KRAS, which is mutated in 10% of all cancers in the USA. And it took my entire life from when KRAS was described as a oncogene, human oncogene, until the first inhibitor was licensed, approved. And these are allosteric inhibitors. So KRAS is a natural allosteric protein. So it has like an on conformation which binds stuff and an off ensemble of conformations which do not. The natural allosteric regulator is the nucleotide binding pocket, so GDP or GTP. And then these covalent inhibitors, of which there are now several, and also non-covalent ones now, these bind in a little pocket next to the nucleotide pocket. So I think for me as a non-pharma person, this is a wonderful story because everyone thought that KRAS was undruggable and had no pockets. Somebody or several labs got stuff to work in one pocket. Now every pharma company on the world has things in that pocket. So it's like sheep after something when it works. So let's make a map of KRAS. So we make a big library which has double mutants in KRAS. And then we select it for binding to different effectors, for example, KRAS binding to RAF1. And then we also measure the effects of the mutations on the folded concentration of KRAS when it's just taken and put into yeast cells by itself. Okay, so this library is about 25,000 mutations. It's in 3 blocks, so it's quite easy to make. And each mutation in the library is there 10 times. So it's there not as a single mutation, but it's there in combination with 9 other mutations. And that gives us too much data so we can constrain the models. So this is what these typical mutation scanning maps look like. So this is just the actual growth rates. So this is the sequence. And then red means bad. So this is all the mutations in KRAS which make it bind less to RAF1. So there's lots of them. But then you look at all the mutations in KRAS which actually just affect its fold stability. And there's also lots of them, okay? And you can really see that many of the changes in binding are caused by changes in abundance. And you can actually just plot these things against each other. So this is just raw data, there's no model. Each one of these is a different genotype, so different mutation in KRAS, double mutants. This is how well they bind to RAF1. And then this is their fold stability, folded concentration. And so you can see when we're model fitting, we do this in a sort of clever way, fitting a biophysical model, but you could also do this in an empirical way, just put a curve through this data. And you can see that, as the protein is destabilized, not much happens because it's saturated, and then the binding falls off. Okay, so that's what typical folding mutations do. And then there's all this other stuff here. These are mutations which have a bigger effect on binding than can be explained by their effect on folding. Okay, so literally you can just measure these residuals and it tells you all the mutations which are changing the binding energy. And so this is binding, but this could be enzymatic activity, this could be DNA binding. You can do lots of different assays here to work this stuff out. Okay, and just to show this works, so the red points here, these are the mutations in the binding interface between KRAS and RAF1. So we would expect a lot of these to affect binding, and you can see that many of these actually have big effects on binding. And then there are all these other black mutations here. These are all the allosteric mutations. Okay, so these are mutations which do not touch RAF1, and many of them have as big effects on the binding to RAF1 as the mutations in the binding interface. Okay, so this is completely raw data with no model fitting, so you can see this stuff in the data. So we do fit models to it, and this means for every mutation we get out a folding energy, which then controls the abundance like this. So life is not linear. And then we get out a folding energy, these are the same, and the binding energy for each mutation, which then controls the binding. Okay, so this is now a 2D function. So these landscapes, these matrices are now not fitness landscapes, they're not measurements of growth rate, these are now in energy units. So this is the effect of every mutation in KRAS on the binding energy and every mutation on the folding energy. And now these are much cleaner and more complete. And we can put them on the structures. So this is just where mutations affect the folding energy. It's in the middle of the protein, kind of obvious. But this is the more interesting one. This is where Now we're just coloring the surface of KRAS, and we're looking at where the mutations have a large effect on the binding energy. In red is bad. And so the binding interface is obvious. Okay, so if we did not have the structure, we'd have worked that out quite easily. But then if you rotate it around, you can see there's the interface, but then there's mutations in other places which are doing interesting things. And so there's this pocket around the back of KRAS where mutations in this pocket actually are affecting the binding energy. So what can we use these maps for? So one thing is just to look at where allosteric sites are, where the active sites are, etc. Here's just an example. Now we're looking at the binding interface of KRAS and what every mutation does in this interface. And so you can really start to understand how the molecular recognition happens. Like here's a tyrosine, and mutating that to any position apart from another aromatic is bad. So that clearly has to be an aromatic to bind RAF1. But then the more interesting stuff is mutations outside the interface. So things which are not touching the target. So this is just a sequence of KRAS. And this is the change in binding energy at each position. So there's 19 dots at each position. The ones in red, these are positions which touch the target. The ones in blue, these are positions which touch GTP or GDP. So these are in the natural control site. And then the ones in yellow, these are the sort of novel, strong allosteric sites. So many mutations in these positions have large effects on the binding energy, and they're not touching the ligand. Okay, and they're a similar magnitude as the interface. So you can put these things on the structure. So again, here I'm just coloring the positions where this is RAF1, this is KRAS. Coloring the positions where mutations have large effects on the binding energy. So you get the interface, you get the nucleotide binding pocket, and then you get some other stuff going across the protein. And if you plot this out as a distance, so this is just the distance away, it's like this distance or 3D, the distance from the interface, and then how much the average of the 19 mutations affect in each position, you get this exponential decay. Okay, so we've seen this for every protein we've done so far. The best predictor of allostery is just how close you are to the active site or the binding site of the protein so far. Okay, so most positions don't change the binding energy on average. Some of them really have quite strong effects, including at a distance, like over here. So looking at the surface again, so this is the nucleotide pocket, it's very red. There's this pocket next to it, which is very red. That's the cytorazib pocket, so that's the sort of control. This is the known, where all the allosteric modulators go. But then there's this other pocket around the back of KRAS, which in our data has mutations. So playing with this pocket with mutations affects the binding energy. The question is, would putting a compound in that pocket also affect the binding energy? So that's the sort of how this would change your decision-making in drug development. So it's a way to rank the pockets by which ones genetics tells you are good pockets to put small molecules in to do particular things to proteins. So just finally on KRAS, KRAS is one of these proteins that binds a huge number of proteins in the cell. So different effectors, RAF kinase, PI3 kinase, other regulators. There's also loads of synthetic antibody-like proteins which bind KRAS that people have made in the world. These are DARPins, these are like synthetic antibodies, and regulators, etc. And so once we've got these libraries, so we've got a library of KRAS, we can just plug in all this other stuff and measure, okay, how do the mutations affect binding to RAF1, but how do they also affect binding to PI3 kinase? How do they affect binding to these inactive state specific antibodies and these active state specific antibodies, etc. So you can just test lots of different things with these assays. And it just means doing the experiments again if the proteins work. So now we have the effective mutations on the stability of KRAS, binding to RAF1, binding to PI3 kinase, binding to RAL-GDS, binding to son-of-7-less, and binding to these DARPins. And you can fit all of this stuff with one model. So it's very flexible the way we do this. And so now you can really, along the sequence, work out where are the positions which are important for binding to SOS1, but are not important for binding to other stuff. Individual mutations in here in the interface, there's loads of mutations which will make KRAS bind stronger to one of its targets and weaker to its other targets. So this is a very quick way to make useful experimental reagents which allow you to tune the specificity, for example. Okay, so you can just put this on the structures of all these things. They all work very nicely, finding the interfaces. So to summarize this stuff, so we've done this on quite a few proteins now. All proteins are allosteric. Okay, it's just a question of how allosteric are they and where are the strong allosteric sites. The effects of mutations on enzymatic activity and binding energies are extremely well correlated with the distance away from the active site. So there's an exponential decay. But it's anisotropic, okay? So this is the interesting question. Why is communication better in some directions than in other directions in proteins? And KRAS itself has 4 surface pockets. They are all allosterically active by mutations, so they are all, we think, decent places to play with small molecules. As you go to bigger proteins, this number increases further. Okay. And then what I haven't shown you is when we start doing these assays again and again and again, so testing binding to different interaction partners, is you can see that it's actually quite easy in a protein to change its specificity or its selectivity by making mutations. So both mutations in the interfaces, but also mutations outside the interfaces can tune specificity. So, okay, here's the problem. We've done one of these oncogenes. Of course, we would like to map the allosteric sites in the rest of them. So this is what our company in Barcelona is doing. They're going after all the oncogenes to try and find allosteric sites in all of those. So if you're interested in that, you should contact them, not me. The more general problem is, of course, we would like to be able to do this for all proteins. So we would like to be able to design allosteric sites for biotechnology. We would like to be able to know where are the allosteric sites on any protein. And so the question is, how much data do we have to produce until we can stop doing the experiments and just predict this stuff? And the other thing we're doing in the lab very actively is developing assays to do this for other activities. Okay, so we have very nice assays now working for GPCRs. So you can make on a GPCR, you have the orthosteric site. You have a lot of other small pockets. Which of those do what on each GPCR? So that's the goal there. For kinases, have a big active site. There are quite a few known allosteric inhibitors of kinases now, but each kinase has many different small pockets on the surface. And so which are the good ones, which are the bad ones? Enzymes as well, you don't always want to just inhibit an enzyme. You want to be able to modulate it, or you want to be able to inhibit the pathogen enzyme, but not the human enzyme, for example. And then one we're very interested in is in transcription factors. So the question is, do transcription factors have nice allosteric pockets that you could potentially drug? And this is just the final example. So this is an allosteric map of a kinase. So this is SARC. This is the ATP binding pocket, and then so you can see the same thing. It's like mutations close to it do a lot, and as you go further away, less. This is the distance. There's this really nice exponential decay as you go away from the pocket, and these are all very strong allosteric sites in SARC. So there's many little fragment binding pockets potentially on the surface, and so now we can say which ones are the better ones and which ones are the less good ones for potentially starting a campaign. So we have to do this bigger. And at least part of the doing it bigger we've partially solved. So all this is about just getting assays to work. So we need to mutate a lot of proteins to measure the effects of mutations on the stability. We need to mutate a lot of proteins to measure their effects on binding and activity and etc. And so this is a kind of pilot project for doing that, where we took 500 human proteins, chopped them down into domains, so little folded units, and then did site saturation mutagenesis of them. So made every mutation in every position and just measured the effect on the folded concentration of the human protein domains inside yeast cells, just to prove we could do it at scale. So this is what this looks like. So each one of these is one protein, the map. And we have, so the published data is 500 of these. We've got another 1,000 finished in the last 2 months. So it's super scalable. You can just keep going through mutating, finding the mutations in human proteins which break their stability, And therefore, you also find for free from this the mutations which break them for other reasons. So if you have a pathogenic mutation that does not affect the stability, then you know it must be affecting something else. So just to quickly give you the take-home message from this, in these domains we've tested so far, 50% of all human pathogenic variants destabilize proteins. Okay, so it's a major mechanism by which mutations affect proteins, but it varies quite a lot across different structural classes of proteins. So some of them, the pathogenic mutations are not destabilizing, they're doing something else. Okay. And then, well, this is more nuanced stuff, but the more of this data we produce, the better we are getting at understanding it and also being able to predict it across different proteins. So this is the general idea, is that the world is pretty good now, but when you have big data sets of building extremely good predictive models, there is one success example in biology that's really good, which is AlphaFold and its mimics. The rest of the world, there are many good examples now. And the real problem is in biology is AlphaFold worked because we had PDB. We had hundreds of thousands of structures, which is an extremely unusual quantitative measurement for biology. And then you have all the sequences of millions, billions of proteins now, which multiple sequence alignments to tell you how these things are evolving. The rest of biology, for example, allosteric, protein stability, enzymatic activities, we do not have PDBs. Okay, there's no protein data bank with this very large, very well calibrated measurements. And so we're super keen on the idea of using genomics to do experiments to make big measurements of how sequence relates to particular activities. So the stuff we're doing is stability, which I've shown you about, amyloids, so protein aggregation, so how do mutations make proteins aggregate? We have quite big data sets on this now. Protein-protein interactions, so how do mutations in interfaces change the affinity of proteins? If we can make a massive data set of that, then we can learn how to predict it. Protein-DNA interactions, the same thing. How does changing the sequence of a protein change its affinity and selectivity for different DNA sites? The one we would really like to do, which is much harder, is to make very big data sets of protein-drug interactions. This is still early ideas. And then all this allosteric mapping, as I said, we're moving it away from protein-protein interactions into enzymes, into receptors, and into transcription factors. Okay, thank you very much. So all of the computational methods were developed by Andre, who's now in AlloX. The experimental methods were developed by this team of people. All of the KRAS data was produced and analyzed by Zhen Chun, who's now in China. And then all of the SARC data and the domainome, so the 500 mutagenesis of 500 human proteins was actually one person doing all the experiments and all the data analysis. Tony. Thank you.